16.3 Practice Guided Problems and Part 5 Beyond the Basics. Now, up till now, we checked the Mendelian inheritance, and we are going to just check something that is not going to follow the Mendelian inheritance, like dominant and recessive alleles, total simple way. So we call it the non-Mendelian inheritance. Number 17 says that we have radishes that the gene for color shows incomplete dominance. So this is a word that we have to stop for a second and highlight that as incomplete dominance. It means that none of the alleles are dominant or recessive, but they are going to produce a kind of a blended phenotype. We will see how it goes. The pure breeding, when you see pure breeding in a text, that means they are homozygous for that trait. Pure breeding red radishes crosses with pure breeding white radishes, and they make purple radishes. So the red radishes at this point are going to contain alleles, either capital R's or lowercase r's. It doesn't really matter, because at this point there's no dominant or recessive allele. Both of the alleles are going to be present in heterozygous organisms, and the white radish contains capital or lowercase w's. When we cross these two purebred or seeds, plants, the red and the white, we notice that all of them going to turn to be capital R, capital W, heterozygous, all of them, and all of the heterozygous at this point is going to be purple. So the genotypic and phenotypic ratios, let's say genotype, all of them 100% is going to be R, W, and the phenotype ratios are going to be 100% purple. Now, at this point, incomplete dominance, think about kind of blending, because both of the colors are going to be expressed, and the red pigment will be mixed with the white pigment, and they are going to just end up in a purple pigment color. Number 18. So certain breeds of cattle show incomplete dominance of coat color. Incomplete dominance, again, the blending at this point. When pure breeding red cattle, pure breed, that means homozygous, breed with pure breeding white cattle, again homozygous for white allele, the offspring show roan coloration. So what we know is the red is RR in our case, the white is WW, and the roan is RW. What are the genotypic and phenotypic ratios of a possible offspring when a roan cow mates with a roan bull? Now, unfortunately, for the roan color, even the scientists do not really agree on whether it's following the incomplete dominance, like a blend between red and white in the coat color, or it can be also co-dominant inheritance. But for our problem question, it really doesn't matter. Co-dominant is a way like one hair will contain red and the other hair will contain white pigments. That's why it adds up to a roan phenotypic colorization. Or it's kind of the mix between red and white as an incomplete dominance. But at that point, it really doesn't matter. What the question is, if we have two heterozygous organisms, we draw the Punnett square for that. We have the R and the W, R and the W. What we will see is the genotypic ratio is the RR to RW to WW is 1. Usually we put the homozygous dominant first, but at this point, because of the incomplete dominance or co-dominance, in this case we will put 1, but the heterozygous will be 2, and the homozygous, the other homozygous, is going to be 1. This is the genotype ratio. The phenotype ratio, this is the genotypic, and phenotypic ratio will be red to roan to white is going to be the same 1 to 2 to 1 in that case. Now, moving on to number 19. It's the blood typing. The blood typing is super interesting because we will encounter with incomplete dominance, actually not. We will encounter with codominance. Both of the alleles are present, plus we will encounter with dominant and recessive alleles too, and as well as multiple alleles. Oh, there's lots of stuff in here. So for multiple alleles, the alleles can be A alleles for blood type, and we shorthand write it with a capital I because it's dominant, and we put a superscript as an A. This is allele for the A. For B allele is capital I and a superscript capital B, shows that they are codominantly, both of them can be present on the red blood cell. And also we have another allele, the O allele, that we will use a lowercase letter I, indicating that this is recessive to both A and B. Now, how does it look like? So here is a red blood cell, and if somebody has a phenotype like A blood phenotype, it means that it has an A marker on its surface, red blood cell surface. If a person is B, contains B blood, that means all of its blood cells contain a B marker on its surface. And if we have a person with O blood, that means it does not have any markers on the red blood cell. Okay, and how does the AB blood person look like? The AB blood person blood cells will contain an A as well as a B marker. Knowing that, let's list the expected genotype and phenotype frequencies of all the potential offsprings of a cross between the woman and a sperm donor. Oh, well, before that, how about we just go and check it out. How is the genotype for A? What would be the genotype for blood A? It can be capital I, A, homozygous dominant, or it can be heterozygous and it still contain an A blood. The B blood, a person with B blood can have genotypes like capital I and B, homozygous dominant for that trait, or heterozygous for that trait. For O blood, the person has to be homozygous recessive, and we have another type of blood, A and B, and that person has to be only codominant for A and B. Now, knowing this, for our problem to solve, we have a man with type AB. So when we draw the Punnett square, we know that the man has A, and I, B, and IA. Alleles to a woman with type B. Type B, so the woman can have homozygous dominant for B or heterozygous. We don't know which one is that. But here's another information for us, and that says her mother has type O blood. So in this case, the mother contributed to the I for the woman, so the woman must be a heterozygous organism for blood type. Now, what we have in here, if we cross these cells, this is the end result. IBI. So I will delete this one a bit, that I would have a place to write. List the expected genotypes. It can be IA, IB, 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 IAI, and IBI. This is the genotypes. For the phenotypes, and the frequency for that 1 to 1 to 1 to 1. And it's also asked the phenotype frequencies. The frequency is going to be AB blood, 1, to B is 2, and the A is 1. So it's 1 to 2 to 1. Good so far? Okay, so number 20. One parent has blood type AB. Here is A, here is B, and the other has type A. We know that it has to have I, and this can be either capital I or lowercase slash. In this case, the cross could yield any blood type except for 0. No O blood, because for the O blood we need to have homozygous recessive for that, and A and B blood type of a parent cannot contribute a lowercase for the offspring. 21. One parent with blood type A and a second parent with blood type B can produce children with what possible blood types? Now, we can have one parent is A and something, and we cross it with a B parent and with something. We don't know at this time whether they are homozygous or heterozygous for that allele, for that trait. Depending on which alleles, we fill it in here on the lines. For the second blank alleles, for both parents, this cross could yield phenotypically to have an A blood, B blood, person AB, and O blood too. So all of the possibilities. Number 22. A woman with type A blood is married to a man with type B blood. They have a child with type O blood. The man claims that the child cannot be his biologically on the basis of blood type. Is he correct? Now, we have the mother is A something, and we cross it with father B something, the alleles, and he is at this point not correct. If both parents contain recessive allele and they are heterozygous, in that case, the offspring really can have the genotype of homozygous recessive, or phenotype-wise a 0 blood offspring with 0 blood. Okay, next one. A woman with type B blood, woman IB something, because we don't know, is going to marry a man with type AB blood. So IA and IB blood. They have a child with a type O blood, so it's going to be II. This is the phenotype is O. The man claims the child cannot be his biologically on the basis of blood. Is he correct? Yes, indeed, he is correct, because in order to have homozygous recessive offspring, both of the parents have to contribute with one allele, recessive allele, to the offspring. And in this case, the male organism does not have any recessive alleles for that. Okay. One parent has another question. One parent has type AB blood and the second parent has type B. the second parent's mother has type O blood. What are possible possibilities of two parents' children? So parent 1 is IA and IB, and we cross with the second parent is IB, but we know that the mother has O blood, so got to have recessive. In this case, what is the outcome of that? It can be AB blood as a phenotype, B phenotype, A phenotype, and that's all. If I just mix them like this, or with this and that. Okay, and the last one is, for now, so achondroplasia, dwarfism, one type of dwarfism, is caused by a dominant gene. That sickness, that abnormality is caused not by a recessive gene allele, but a dominant allele, dominant gene. Homozygous achondroplasia results in the death of the embryo. So it means that, okay, let's write that down, what we know. Capital A stands for achondroplasia. This is the achondroplasia gene, defected gene, and lowercase is normal gene. We know if somebody is homozygous for the A gene, in that case, that person is not going to make, it will die in utero as an embryonic age. Two parents, so what we know, anybody with achondroplasia, dwarfism, they are heterozygous. They must be heterozygous. They have to have one normal gene and only one dominant achondroplasic gene. So two parents, both with dwarfism, have a child. Draw it out, the Punnett square. And both of the parents are heterozygous, because if they would be homozygous dominant, they would not be alive. They have a child. Let's do the crossing. What is the expected ratio of a dwarf to non-dwarf offspring? Well, since having two dominant alleles in here, and we said dominant will not make, so they will not get born, is embryonic lethal. The parents both heterozygous parents are going to produce a 2 dwarf and 1 normal height offspring. So the phenotypic ratio is 2 to 1 in this case. Thank you.